Moving on to the second prize for the poster competition. And I introduce Dr. Is it Dr. Gan is going to present, yes? So the title of the poster is a multicenter randomized double-blinded Propofol-Controlled phase 3 clinical study to evaluate the efficacy and safety of HSK3486 sorry, 86, sorry, injectable emulsion for induction of general anesthesia in adults undergoing elective surgery. In adults.
Can you hear me? That's better. As I said, this meeting is always a fun meeting, and I would not miss this meeting for anything. And what a treat to hear Talmage for that lecture. He always can put it at a very simplistic level that at least I can understand it. And just looking out the number of past presidents and of the society and friends, it's great to be able to get together after all, almost seems like a decade long before COVID. Anyway, I'm presenting this poster as part of the investigator and also help the company to design the protocol. So what this, just give you a bit of background. We all know Propofol. Propofol has been used for many years. It's a great induction agent, but it does have some adverse effects and effects that we perhaps do not want to see. You know, hemodynamic instability, pain on injection in particular, respiratory depression. So this compound HSK3486 is similar to, is an induction agent, very similar chemical moiety to Propofol, but potentially may have a more desirable effects and less side effects. So that was really the intent of this study. Is a phase 3 clinical studies to look at efficacy as well as safety of the drug. And based on the phase 1 and phase 2 studies, that is when we saw some of the advantages of this compound compared to Propofol. So the primary objective is to demonstrate non-inferiority of HSK3486 compared to Propofol. And the way we define the margin is the non-inferiority margin of -8%. The design, 255 participant randomized controlled trial undergoing general anesthesia, ASA 1 to 4, over 18 years old, BMI above 18. And so randomized into 2 groups, HSK3486 and Propofol. The design is that the study drugs 0.4 milligrams IV titration in induction over 30 seconds. And then a minute later, if there is no desired effects measured by MOASS, as we discussed today a lot. And then if that doesn't reach MOASS of 0 or 1, a second dose of 0.2 milligrams per kilo will then be given. And then MOASS is continually being evaluated every 30 seconds for 2 minutes. If then still MOASS is more than either 0 or 1, then the rescue drug will be given and the patient will be considered as failure. Likewise, in the Propofol group, similarly 2 milligrams per kilo, IV induction over 30 seconds. And then a minute later, if MOASS is not less than or equals to 1, additional dose was given of 1 milligrams per kilo, and likewise 2 minutes later, then obviously Propofol will be used as a rescue. Essentially, the primary outcome is to, for success, is defined as MOASS 1 or less than 1, 1 with the maximum of 1 top-up dose, and without any rescue drug. Secondary endpoint, injection site pain, as well as successful induction as defined as maintained desired depth of anesthesia. And of course, the safety endpoints were also collected as well. So that is the demographics. Essentially, is a 2-to-1 randomization. You can see the number of patients in 3486 is about twice the number of propofol patients, but the demographics are very similar between the two groups. And just to present you the primary endpoint, essentially there was no difference in the efficacy between HSK3486 and propofol, achieving 97 and 97.6% successful induction, respectively. And again, is within the realms of the non-inferiority of -8%. So this drug was demonstrated to be non-inferior to propofol within that lower bound of 8%. Secondary endpoint, we did see a significant difference in terms of pain on induction. So the pain on induction was assessed using a numerical rating score that the patient were being assessed every 15 seconds as you're doing an induction period. And you can see here on the top bar, the score of above 1 or above 1 is 77% for propofol, 18% for HSK3486. And more severe pain score over 4 is about 61% and 6%. And both are statistically significant. So pain on induction was less with HSK3486. And there is no difference between study drug and propofol in terms of maintaining the depth of anesthesia. Safety endpoint, there were higher number of study-related adverse events in the propofol group, 53% compared to 22% in the HSK3486 group. So in conclusion, the study met the primary endpoint of non-inferiority between propofol and HSK3486. At successful induction anesthesia was associated with less pain on injection than propofol, and the overall incidence of study-related adverse event was numerically higher in the propofol group compared to HSK3486. Thank you very much.